Okay. Okay. This video is the sixth tutorial for the course I Think Biology, which is an NPTEL course offered by multiple professors from the Azim Premji University. I am Geetika Agarwal. I'm a PhD scholar at the Center for Ecological Sciences in IISc Bangalore, and I will be taking this problem solving session for you guys today. Okay, I will give you a brief background of what was covered, a brief summary of what was covered during this week, all the major concepts that were covered during the week, and then we will have a small Q&A session to align you guys towards what happened during the week and to help you with your exam and answer these questions better. Okay, so the mostly what they talked about this week, one of the lectures was entirely about bead microscopy and how it's conducted. Give me, yeah. So, uh, yeah. So the major concepts that were covered during the week included bead microscopy, in which you learn how you can use a glass bead as a microscope to look at small objects. Then it talked about molecular cloning and how this works and what is recombinant DNA, what are restriction enzymes, plasmids, what is the technique of gel electrophoresis, what is PCR, and talked a little bit about BT cotton. Okay, so moving on to the first thing of bead microscopy, let me give you a brief history of microscopes in general. First of all, the humans started using lenses or magnifying glasses, and the first documented use come from around 1400s to look at some small objects. The compound microscope, which has two separate lenses like the objective and the eyepiece, that was probably invented by Galileo Galilei, who you might know for his invention of the telescope. But when he inverted his telescope, he figured out that it can be used to magnify small objects and then look at them. So that's why it could be used as a microscope. He did this in 1609. And then comes Robert Hooke in 1665, who designed a compound microscope and used it to observe cells. So, as you might remember him from our previous tutorial in which we talked about cells, which are small compartments. So, Robert Hooke was the one who discovered the presence of cells and that they are the unit of life when he was observing a dead bark under his microscope. So, that is how the microscope came into being. Now, looking further, there is Anton von Leeuwenhoek, as you can see in this image, and he used a bead microscope to observe textile fabrics. So he was mostly into textiles, and he had a business of fabrics. And you can look at this at that time. This was the bead microscope. This is the small glass bead, and he used this to not only observe textile fabrics, he also observed some biological specimens. And in this image, you can see his illustrations of various biological specimens that he took a look at. For example, you can see here there are these different kinds of seeds and like small plants and things like that. He's also called the father of bead microscopy for the same reason that he was the one to use the bead microscope to observe small specimens. Then there is this person called Robert Brown, and he discovered nucleus in an onion cell using a simple bead microscope. And that was around 1820. So you can see here what he was seeing through his bead microscope. These are different onion cells. These are the stomata, as you might be aware, with the two guard cells. And in the onion peel, you can see here these small nucleus, these small round structures within each cell. So he discovered the presence of the nucleus. Now, looking at the simple microscope and how that functions. So, a simple microscope is basically just a convex lens. You might be aware about this from your physics high school lectures that a lens with rounded edges is like a convex lens, and the other one is a concave lens. And when you look through a convex lens, if you see a small object, for example, in this ray diagram, A is the object, and then the image is formed somewhere further behind the object, and it is the a prime. So it gives you a magnified image of the same object. So convex lens is used to view a small object and make an enlarged image of it. How is the magnification calculated? The formula is m is equal to 1 plus d by f, where d is the least distance of distinct vision. So the least distance at which you can distinctly see objects. F is the focal length of the lens. So focal length means where it is going to focus the image. So here you can make out that magnification is basically inversely proportional to the focal length. So a length that has a smaller focal length will have a higher magnification, and a length that has a higher focal length will have a smaller magnification. Resolution is calculated as by the formula lambda by 2 into n sine theta. Here, lambda, as you might be aware, is the wavelength of light. N is the refractive index of the medium from which the lens is made. So it's the material of the lens. So lambda, so N is the refractive index of the medium. That is basically the material of the lens. And then theta is half of the lens's angular aperture. So looking at the lens's angular aperture, how much light it, like what is the aperture of this lens, how much light can go in, half of that is theta. And that is how you calculate the resolution. So one thing from here is clear, that larger the aperture means the resolution will be better. Okay, if the aperture is large, it means the resolution will be better, but the magnification will be smaller. So basically, it's a trade-off between a resolution and aperture. So if you all might be using cameras, you might already be aware of this. If your lens has a larger aperture, the resolution of your image is going to be better, but the magnification of it is going to be lesser. So it's not going to be good at magnifying the image. Okay. So this was some things about the simple microscope. Coming to a compound microscope and its ray diagram. So this is what the compound microscope's ray diagram looks like. Basically, in this, what happens is two lenses. You can see here O and E, objective and the eyepiece. Eyepiece is the lens that is near to the eye, and objective is the lens that's near to the object that you're trying to look at. So that's pretty straightforward. And so these two lenses, they're kept along the same focal plane. So you can see here that the focal plane is same of the objective and of the eyepiece both. And here you can see that objective is will first make an image of the object. So this object, its image will be made here somewhere by the objective. Okay, A image is A prime, and of this image the eyepiece will make another image which is A double prime, and which will be even larger than the one made by the objective. So this here, this much part, which is the objective and the image, you can think of it as the, this here is what a simple microscope does, right? And from this so from this, from the object at A, so this is the object upright A, its image is made here at this point A prime, and from A prime the rays travel through the eyepiece, they reach the eye, and you see an image A double prime, which now you can see is much bigger than A prime itself. So the use of two lenses is giving you a higher magnification, it's producing a bigger image to be observed. So this is why compound microscope is better than the simple microscope. But as you might be like able to guess by now, that this needs a lot of proper positioning of both the objective and the eyepiece, and there are many more things here, that many much higher level of engineering that comes into play when you look at a compound microscope. Magnification of the compound microscope is basically the product of the magnification of the objective and the magnification of the eyepiece, and therefore it is inversely proportional to the product of the focal length of the objective and the eyepiece. And compound lenses, they have they you face issues when you're looking at compound microscopes, because there is an increased amount of chromatic and spherical aberration. I'll explain to you with a diagram what is chromatic and spherical aberration. So basically, chromatic aberration is when the wavelengths of different colors, they are focusing at different points, and spherical aberration is when the light rays, they get focused at different points. So as you can see in this image, this is spherical aberration. Here light rays are traveling, and when they pass through these lens, they're not able to focus at the same point. So if you think of this as the focal point, the images, all the rays are not going through the same point. There are two foci here. So that leads to some blurring of the image, because not all rays are meeting at the same point. So this is called spherical aberration. And something similar for chromatic aberration is when you have different, so as you might be aware, different colored light waves have different wavelengths. So when these different wavelengths pass through the lens, they might get refracted in different ways, because refraction depends on the wavelength of the light. And then these different colors might be focusing at different points. You can see here red is focusing the first, then comes the focal length of green, and then is the focal length of the blue rays. So that is why you'll see some different colors in your image. So that leads to a little bit of chromatic aberration. So these are issues with using a compound microscope. So coming to a simple, straightforward comparison between a simple and a compound microscope, first thing is that, as you are now aware, a simple microscope only has one lens. So it is pretty simple or pretty easy to assemble because there is only one single lens. Compound microscope has two lenses, and the alignment of there is very crucial. As we discussed, they should both have the same focal plane. So it's very crucial how you align these two lenses so that you can get some magnification. Otherwise, you'll not be able to get the correct magnification and focus in your image. In a simple microscope, a small bead is needed for a high magnification. In a compound microscope, there are two lenses, and the total magnification is the product of the magnification of each lens. So each lens can actually be relatively large. So why we are saying this, as we discussed, like aperture is inversely proportional to the magnification. So for getting a higher magnification, you need a very small bead so that the aperture is small. So aperture is basically the area through which the light travels. Matlab, how much area is available for the light to travel through it. So that is the aperture. So if you have a small bead, so the area through which the light comes into this bead is small, but if you have a large bead, the area through which the light can come inside is much gets much larger, right? So that is about basically. So if you have a much smaller bead, it will give you a bigger magnification. When it comes to a compound microscope, you can actually use some pretty decent sized or larger sized lenses, which are actually easy to manufacture. They're cheaper, and so you can use these larger sized ones because the total magnification is anyway going to be a product of magnification of each lens. In simple microscope, you have very limited spherical and chromatic aberrations, but there is very high distortion. So the image is very distorted. So you will not see like a plain image like this of a rectangle might appear like this with bulging sides on the outsides of it, or it might appear with concave sides. Okay, so this is distortion. So this distortion might happen when you look at an image through a simple microscope. Here in compound microscope, you have significant amounts of spherical and chromatic aberrations. Now, simple microscope is actually inconvenient and it's tedious to use because you have to keep it very close to your eyes, and then the object also should be very close to the microscope. So that makes it difficult to use. But the compound microscope is convenient to use. On the other hand, simple microscope is very affordable and robust. Why is it robust? Because it's just a little glass bead. So it's, of course, it's very robust, and it's very affordable because it's just a small bead. But when you look at a compound microscope, there are so many things, so many parts that come into play, and you have to align everything in a particular focal plane. So it becomes that makes the entire engineering process quite expensive, and the and it's quite fragile, so it can be easily broken. That is why you'll only find compound microscopes in like facilities in your colleges where they are like kept locked, and they can only be used by certain authorized individuals who have been trained to use a compound microscope. Not everybody will be allowed to use a compound microscope without supervision. So you need to take good care of a compound microscope. Moving on to bead microscopy, which this lecture was about. So, in bead microscopy, basically, they are DIY. You can make them yourself. They're quite cheap and they are robust. As we discussed, it's just a glass bead. And the DIY microscopes that were made by this team who was giving the lecture is actually inspired by the fold scope. And fold scope was made by this professor, uh, and I'll play a video of his, so as to get a better explanation of. So it was made by this professor whose name is Manu Prakash, and he's from Stanford. And here I'll play this video in which he's talking a little bit about his fold scope. You can go and take a look at the entire video to get a better idea of how he actually thought of the fold scope and came about with this invention. So, but it's basically origami based microscope in which different shapes of paper come together and make this microscope. So, let's listen to Professor Manu Prakash.
Cutting process on a piece of paper. Here is what we call as a sample holding stage. Here is the optics stage with the optics module actually put in. And thirdly, we have an illumination stage which has the light source, a watch battery, a switch, and an LED with a condenser lens on the other side. Each one of these stages folds and interweaves together to form an individual microscope that looks something like this. It takes maybe of the order of 2 to 3 minutes to fold this object in, and folding gives you a fully functional instrument. There is a tiny little slot right here that allows you to put a glass slide. So I'm just going to do a quick test to show you how to put the sample in. So at this point, the sample has been registered to the slide, and you can image it very directly to your eye either by looking right here or essentially using this instrument in a projection mode where the image is projected outwards. The way to use the instrument is by both your hands, where you move your hands to change your field of view, and the tension in your hand gives you focusing. So just slight tension in my hand allows me to move my focal plane up and down to actually focus tightly on the sample that I care about. There is a lot of number of parts.
So this was a little bit of about Professor Manan Prakash and its full scope. Basically, as I explained, there are these three parts: one which is the sample holding stage, one which actually contains the lens, and one which has the LED to provide the light source to view the image. And then you can basically, they give you these cutouts, you can cut them out and fold them together, and that's why it's called a fold scope. So that is what the inspiration was for this normal bead microscope being used by yeah, these bead microscope being used by ISPF and Think Tank, which are actually NGOs that um, associate with schools to teach kids about how to using these things. So basically, in bead microscopy, what all is required is that they use this ice cream stick. In the ice cream stick, they made these two holes where they attach their glass beads, and then they were secured by foam and then by tape. And then there is this microscope onion cell. So the onion peel, they secured it on a glass slide, and they basically dyed it with saffron to look at the cells. And then you can look at these, you can basically place the slide pretty close to the right next to the ice cream stick containing the glass bead, and you can even actually put a grid on top of the glass bead to get an idea of what is the size of the different cells. So you can paste a grid on top of the glass bead, and then you can put it very close to the onion peel and then put it in the light, say against the sunlight, and then you'll see what is happening inside, or what how the cell structure is of that onion peel. So that was all about bead microscopy and how it's like very new and very affordable way of teaching kids about what are microscopes and looking at some small objects, and building interest in biology and microscopy. Now, coming to the next concept, which is of molecular cloning. So, we will discuss what is the molecular cloning technique and what are the different components that go into this technique. So, first of all, molecular cloning, it's basically a technique where you are expressing a target gene which belongs to a foreign organism, and you want to express this target gene into your host organism. For example, we are talking about some antibiotic resistance which exists in some bacteria, but you want it to be expressed in some other bacteria as well. So your foreign organism is is that bacteria which already has the antibiotic resistance, and you wanna take that antibiotic resistance and now put it into your host bacteria. So it's a way in which you want to express one target gene, or a target gene which belongs to a foreign organism, and you want to express it in your host organism. And then you would like to create several copies of the target gene, and which will happen by multiplying the host organism. Because your host organism now already has the target gene, you can leave it to multiply and to go generation after generation, and the target gene will also get multiplied along with the host organism. So it's basically production of multiple copies of recombinant DNA by the host organisms. So this is what this technique looks like. I'll give you a brief step by step of the technique, and then I will come to explaining what is plasmid and what are these enzymes. So basically, first of all, in this bacteria, you can see there is this plasmid and there is this chromosomal DNA. So this plasmid is isolated from the bacteria. Into the plasmid, it is cut with an enzyme, and then here from another cell, you isolate the DNA, and this red portion is your gene of interest, which you want to multiply. That's called the target gene. Okay, so what is happening here is this is the plasmid. It is cut with some enzyme, so it has now an open end. And here, this is the gene of interest in red color. and you're trying to multiply this target gene, which are called as target gene. So this target gene, then you paste this target gene into the um, plasmid. So you ligate it with the plasmid, and now it is the one shown in the blue. Okay, and then you put this recombinant plasmid into your bacteria. So put bacterium into bacterial cell by transformation, and then you allow these cells to multiply. So that is basically molecular cloning. Another way to see is the colony is grown into a large culture. The bacteria are induced to express the gene, so the gene is transcribed into the mRNA. So from these cells you basically so this is the recombinant plasmid, as we see here also. It has antibiotic resistance gene here in green. So what you do is you then put the bacteria on an antibiotic plate. So basically the substrate contains the plate. So all of the bacteria which is without the plasmid, which do not have the plasmid or which did not get the gene properly, all of them will which do not have the plasmid, basically all of them will die, and only the cells that have the plasmid will grow on this thing. And then from this bacterial colony, if you want to express your gene of interest, your target gene, for example, say it's a gene that produces insulin and you want to harvest large amounts of insulin, so you can then grow that colony of bacteria which is containing that particular plasmid, and then they are induced to express the gene of interest that you require. And so that protein will be produced in large quantities. So that is all about molecular cloning. Now we will look at step by step how this is happening. First of all, what is recombinant DNA? We've been talking about recombinant DNA and how we want to how this plasmid, which is ligated with the target gene, is called recombinant DNA. So, recombinant DNA basically, as the term suggests, is a DNA molecule which contains DNA from two different sources. So, it has DNA from either two different species or two chemically synthesized DNA molecules. For example, this was the host plasmid, which had no foreign DNA in it. Then, at the site of cleavage, some of the enzymes come and cut it, and now it is open here. Here, another the specific genes or the target genes that you want to amplify from another DNA source, they come and bind here. And now this obviously becomes a recombinant plasmid DNA because it has DNA from two different sources. This is from the organism from where you harvested the plasmid. So this is the host plasmid from the organism from which you got it, and this portion is the target gene which comes from some other organism altogether. So basically, DNA is coming from two different sources. That's why it's called recombinant DNA. I hope that is clear. Now, talking about transgenic or genetically modified organisms, you might already be aware of the term genetically modified organism because it's very commonly used in mostly in terms of the food that we consume, agriculture, produce. So they all talk about GMOs and how the plants that we are consuming or the vegetables that we're consuming are all genetically modified and they might not be good for us and things like that. So one way to look at genetically modified organism is basically it's an organism which has now acquired foreign genes artificially. So for example, in this diagram you can see there is this some bacteria from which we basically put like that plasmid, we put that gene into this that plasmid into this bacteria, into this plant cell, sorry, we put the plasmid which is containing or we put that bacterial portion which contains the target gene into the plant cell's chromosome so that it is expressed, and this plant is now transgenic because it contains the particular gene that we wanted it to contain. So, and an example, as I said already, these days many crops are genetically modified in order to provide them with resistance to pests, to improve the yields, to increase the fruit sizes, to increase the seed sizes, and things like that. So basically, genetically modified organism is nothing but an organism that has one or more foreign genes which have been introduced into it artificially. So here, this bacteria's gene of toxicity, the Bt toxin, it is introduced into the plant, and that is happening artificially because it is happening because of human intervention. Humans are actually at play here. Researchers are isolating this gene from the bacteria and introducing it into the plant. Okay. Moving on to restriction enzymes. What are restriction enzymes? So basically, we were talking about how the plasmid is cut by some enzymes, and then there is open space where where the target gene can go and ligate or go and join, right? So those enzymes are actually restriction enzymes. So these are proteins that cut DNA at certain sequence specific sites. So sequence specific means that these restriction enzymes or these proteins are capable of recognizing the DNA sequence, and they go through the DNA sequence and they only cut at some specific sites at which they are active. So they can either be endonucleases or they can be exonucleases. So you call a restriction enzyme an endonuclease if it is making a cut within the DNA strand. So if you have a stretch of DNA and the restriction enzyme is cutting somewhere in the middle of it, then you call it a endonuclease. Okay. But you call it an exonuclease if you have a DNA strand and the restriction enzyme is making a cut at the edge of it. So it's cutting off the tip of the DNA, for example. And now you just have this much left. So that is when it's called a exonuclease. So if it's trimming the ends of the strand, they are basically isolated from bacteria. So they're taken out from bacteria, these restriction enzymes. So you might have a question about why are these restriction enzymes present in bacteria. So in bacteria, these restriction enzymes play an important role in protecting the bacterial cells against bacteriophages. Bacteriophages are actually viruses that attack these bacteria. So what the viruses do is they try to inject their own DNA or their own RNA into the bacterial cell, and then they make the bacterial cell multiply the viral DNA and also produce the viral coat so that the virus gets multiplied at the expense of the bacteria. So these restriction enzymes, they protect the bacteria by actually cutting down the virus's DNA. So when a virus injects its DNA into the bacteria, these restriction enzymes, they snip the virus's DNA, and now it can no longer impact the bacteria. Another thing to keep in mind is methyl transferases. Methyl transferases are actually okay, I'll just write it out for you guys. Methyl transferases. Methyl transferases are actually molecules that, as the term suggests, they transfer methyl groups through the DNA. So what happens is when I'm sorry for the bad handwriting, but I'm trying my best to write it out for you. Methyl transferases. Actually, I'm sorry about this. So basically, I will type it out in the chat box. What are methyl transferases? So methyl transferase. So methyl transferase are actually these molecules which can transfer methyl groups to the DNA of the bacteria itself, so that these restriction enzymes are unable to affect the bacteria's own chromosomal DNA. So you these two compounds or these two enzymes, um, work in combination with each other. They work in combination with each other to protect the bacteria against the infection by a virus, against the bacteriophages. So that is what are restriction enzymes. So they are isolated from bacteria. So one of the bacteria restriction enzymes is called EcoR1. It is isolated from Escherichia coli. That's why the term eco. And the strain of this Escherichia coli is R. That's why R. And this is the first such enzyme to be isolated. So that is why the I comes from. EcoR1 isolated from E. coli, strain R, and first such enzyme to be isolated. BAM-H1 comes from Bacillus amyloliquefaciens, strain H, and this is again the first such enzyme to be isolated. That's why it's the BAM-H1. So you can see here EcoR1, it's making a cut here. So there is a sequence called which is GAATTC, and if you read it from the other side, it's still GAATTC. So these are called palindromic sequences. Whether so from 5 dash n to 3 dash n, if you read them from 5 dash to 3 dash, so they will read the same on both the strands. How other sequences are not palindromic, you can see here this sequence reads GCTGA on this strand, but on this strand it reads TCAGC, which is not the same as that. So these are not palindromic, but this EcoR1, this reads the palindromic sequence of GAATTC. On the other side also it's GAATTC, and it makes a cut between the G and the A. So as you can see the scissors, it makes a cut here at the G and the A, and it gives you these outcomes. Okay, so more than 3,600 endonucleases they have been identified so far, and they can cleave more than 250 specific sites within the DNA. Moving on to the output of restriction enzymes, as we discussed, can create these cuts, and they can lead to these different outputs of either sticky ends or blunt. blunt ends, actually, blunt ends. So they recognize palindromic sequences, as I already told you, palindromic sequences which read the same from 5 dash n to 3 dash n on both strands. So this is a palindromic sequence GAC, GTC, GAC, GTC, CTT, AAG. They read the same from 5 dash to 3 dash on both the strands. Okay, so these enzymes, they actually recognize these palindromic sequences, and the cuts that are made by the restriction enzymes, they might lead to blunt ends if there are no DNA overhangs. So it's a clean cut, there is no extra nucleotides on any side, so that's a blunt end. Or sticky ends if there are these extra nucleotides present which are hanging from the DNA. Sticky ends are basically these DNA overhangs. So these are the extra sites, or the extra nucleotides that are hanging after the cut is made. Okay, and these sticky ends are very important because that is where your target gene will come and ligate. So your target gene will also have a sequence which is complementary to this. So basically your target gene will now have ACGT again. So your target gene, so basically when you use restriction enzymes, you cut the plasmid and the target genes with the same restriction enzymes, so you get the um, complementary nucleotides, or the complementary sticky ends, so that they can come and ligate together. Okay. Coming to plasmids, what are plasmids? We have been talking about plasmids and how they are important for molecular cloning and how they are used. What exactly are plasmids? So plasmids are actually extra chromosomal DNA molecules. You can see here, this is a bacterial cell, and this is the bacterial nucleoid. Basically, it does not have a nuclear membrane, but it's a region where the bacterial chromosome is present. So it's the circular chromosome. The bacterial chromosomal DNA is here, but there are these extra DNA molecules which are called plasmids. So that's why extra chromosomal DNA molecules, and they're pretty small in size compared to the chromosomal DNA. They are circular. So they are circular. They are double stranded. They can either be coiled or linear. So here it's linear because it's not coiled around. So there are circular double stranded DNA molecules, and they can replicate independent of the host chromosome. So, for example, this bacterial chromosome will replicate only when binary fission is happening, only when the bacteria itself is duplicating. But that is not the case for plasmids. Plasmids, they replicate independent of the host chromosome, so they don't need to um, be stuck with replicating only when the DNA, when the bacteria itself is multiplying. They can replicate independent of the bacterial chromosome. They are present in bacteria, archaea, and sometimes in eukaryotes, such as in some yeasts. And they vary in size from 1 kilo base pairs to 1000 kilo base pairs. So kilo base pairs are basically 1010 to the power 3 base pairs. So 1000 base pairs. So they can vary from 10 to the power 3 base pairs to 10 to the power 6 base pairs, right? And they often confer benefits to the host cells. So these plasmids, when they're present in the bacteria, they're actually give benefits to the host cells because they're mostly carrying genes that offer or that provide antibiotic resistance. So if a bacteria has genes for antibiotic resistance, you can treat it with some antibiotics, it still not die. So it can give it, so it can either provide antibiotic resistance genes, or it can be carrying toxin genes which will impact the predators of these bacteria, which will not be able to consume them. So these bacteria can become toxic or improved virulence. So these plasmids, they carry genes for antibiotic resistance, toxin production, or virulence. Okay, so if the bacteria is actually attacking some organisms, its virulence will be higher if or it will be highly virulent if it has these plasmids. Okay, so this is all about plasmids, and let's look at the structure of the plasmid. So now we know it's circular DNA. Let's take a closer look of what all regions are present in a plasmid. So as you can see in this image, a plasmid has, starting with the ori, it's the origin of replication, which you, as you might be aware, it's actually present in DNA molecules. Also because that is what is required, or where the DNA polymerase will come and attach and start replicating the DNA. It has a selectable marker, an antibiotic resistance gene, promoted region, some restriction digestion sites, and the gene of interest is here. So let's look at each of these. So RE sites, which can also be called the cloning site, it actually has these restriction sites for the restriction enzymes to cut and for the gene of interest to be ligated. So the cloning site actually has these restriction sites as mentioned here. And here the restriction digestion will happen. The restriction enzymes will cut it, the endonucleases, and the gene of interest will come and ligate. And gene of interest is the target gene which you get from the foreign molecule from where you want that gene. Okay. That's the gene of interest, which is the one you want to introduce into your host organism. Origin of replication, as I explained, DNA polymerase requires an origin of replications to start the duplication of DNA. Selectable marker, basically, it what it does it can be giving you a color, such as there is a selectable marker called the blue and white marker, where if the plasmid enters into the cell and it's successful, it gives you blue colonies, and if the colonies are white in color, that means it is unsuccessful colony cloning. Why? Because this selectable marker produces blue color. So if the plasmid has entered the bacteria, it will give you a blue color. If the plasmid has not entered, the bacteria will just be white color. Then there is this antibiotic resistance gene. It also helps you choose for the bacteria which can grow on the antibiotic plate. So when you are making these petri dishes, you actually add this some antibiotic to the substrate that you're adding. Okay, so to the substrate on which you are growing the bacteria, in that you add an antibiotic. So what happens is the plasmid contains an antibiotic resistance. As I said, plasmids confer antibiotic resistance. So the same antibiotic you add to the substrate, and when you grow now bacteria on the plate, only those bacteria will grow which actually have successful cloning of the plasmid, which actually have taken up the plasmid successfully, and they're expressing it. Only that those bacteria will be able to grow, which in which the plasmid has successfully inserted, because it makes logical sense. They can grow on the antibiotic plate only if they have antibiotic resistance gene. So, and since this is only present in the plasmid, they will grow only if the plasmid has successfully entered the cell. Promoter region, as we have discussed earlier during transcription, during central dogma tutorial, it controls gene expression where the RNA polymerase would bind. So promoter region is where the RNA polymerase comes and binds, and it will control the expression of genes, right? Okay, so this is all about the structure of the plasmid. Moving forward to gel electrophoresis. Gel electrophoresis is a lab technique. It separates if you have a mixture of biomolecules, it will separate individual biomolecules from this mixture. And how you do it is you apply an electric field in order to do this. And this electric field causes the biomolecules to migrate in a gel matrix, so which means you also have a gel matrix involved. And this gel matrix is usually made up of agarose. So it's a technique to separate individual biomolecules from a mixture. So you can see here in this image, so there is this gel matrix marked in yellow. There are these wells. This is the solution or the buffer solution in which you dip the gel, and then these slots or wells, they contain the DNA, and then you apply current. There is a negative electrode cathode and there is a positive electrode anode, and you mostly place it in such a way so that the wells are facing the negative electrode. And since DNA is negatively charged, when you apply the current, the DNA will start moving towards the positive electrode. And here you can see how different sized DNA or molecules will separate from each other. This is a real image of a real life agarose gel. This is what it looks like. It's like a jelly. It's a gel, and these are what the wells look like, in which you load the DNA containing dye. And then when you view them in UV light, you see these brightly colored bands for different DNAs. So, giving you a brief overview of gel electrophoresis, you basically first mix agarose in the buffer. So, you mix it and you boil it in a microwave so that it's free of any other bacteria. But you basically boil it and you convert it into a proper transparent liquid. When it's properly done, you cool the mixture to about 65 degrees Celsius, and then you pour it into a mold. Make sure this mold already has the comb inserted in it, and this comb is what will lead to making of the wells. And this comb can be of different sizes based on how much DNA sample you want to put in your gel electrophoresis. So you put this comb and then you pour the mixture. Then you wait for this to solidify into an agarose gel. Then you remove these sides which were preventing the spillage, and now you have this finished gel. So the gel is now solidified at room temperature. It is solid only, and the comb is removed, and now you can see that the wells have been very well formed. So this is the finished gel. Now what you do is you take your DNA samples and you add the dye to these and mix them properly. Then you load this sample using a micropipette. You load them into these different wells, and then, as discussed, you run them through the machine, which is connected to the negative and the positive currents. And when the current is run, the DNA will run through the gel, and you can see here, this is how it will look like. And you can see that there will be a ladder. So there is something called the DNA ladder. Here you can see in this image, this is the DNA ladder. So in this, the different, so in this, The researcher is already aware of the size of the different bands. So you already know that, okay, the band that goes here is 100 base pairs, then 200 base pairs, then 300, then 500. The researcher is already aware of that. So when you run the gel, you compare where your strand is coming at, and then you can know what is the size of your strand. Okay. Moving on to another lab technique, which is called the polymerase chain reaction, or PCR. PCR was actually developed by Kerry Mullis in 1983. And Kerry Mullis was actually working with the Cetus Corporation, and he developed this technique. So the thought of actually amplifying DNA already existed, but there was not enough technology to work on it. So then Kerry Mullis thought that, okay, if we have small primers which are complementary to the pieces of DNA that we want to amplify, then we can actually bind them to the DNA and let the DNA polymerase amplify it. And then we can repeat the cycle of this multiple times to get to amplify the DNA or get multiple copies of our DNA. So basically, polymerase chain reaction is used to amplify or create multiple copies of a sample DNA molecule. Right. And the different steps involved are: you start with a template DNA. So you have a template DNA which you want to multiply. Then you add to that a mixture of DNTPs, which is the raw material, the nucleotide triphosphates. Then you add the primers as well, which will actually attach the edge of the template DNA and start multiplication. So primers are these small pieces of DNA which can be either synthetically generated. So they are these small pieces of the DNA which will then bind to your template DNA. So if this is your template DNA, like this, so the primers are actually small pieces which this much and this much, and one of them will start moving in this direction, other one will start multiplying in this direction. So that is how PCR works. So you get primers and you get DNA polymerase, in this case actually Taq polymerase, because it is Taq polymerase is named so. It's a DNA polymerase which is actually isolated from this bacteria called Thermus aquaticus. And why do we use this particular polymerase, Thermus, which is extracted from Thermus aquaticus? This is the spelling of the bacterial name. So Thermus aquaticus is the bacteria from which you extract Taq polymerase, and you do so because this bacteria, it can withstand very high temperatures. It's found in hot springs. So the Taq polymerase can also withstand very high temperatures and can perform DNA duplication even at high temperatures. So because in PCR involves increase of temperature, decrease of temperature. So you need the polymerase to actually be able to withstand these high temperatures. That is why we use Taq polymerase. Then you run the cycles of denaturation, annealing, and extinction, which I will explain in the next slide. And then you repeat these running of these cycles again and again till you get the amount of DNA that you're actually looking for. So these are the steps involved in a PCR. So first of all, in the first step, in denaturation, so this is one cycle of PCR in which there are three steps: denaturation, annealing, and synthesizing or extension. And then you keep repeating this cycle again and again. So in this one cycle, the first step of denaturation is when you increase the temperature to about 95 degrees Celsius. And what happens in denaturation is this double helical structure of the DNA, this double stranded DNA, it actually gets separated into two single strands. So these are the two single strands. And as we discussed, we add the DNA primers, we add Taq polymerase, and we add these DNTPs. Now, the second step of annealing starts. So you reduce the temperature and the primers start working. The primers, they anneal, they attach themselves to the DNA. So if the temperature is high, the DNA strands will stay separated. But when the temperature is decreased, these primers will go and attach to the DNA. And then in the last step, which is synthesizing, which happens again at a little higher temperature of 72 degrees Celsius, this Taq polymerase will come and attach, and it will start multiplying the DNA in two different directions, right? So from 5 dash to 3 dash, it adds the DNTPs. So the Taq polymerase will multiply the new strands. So once this is done, so from one DNA, from one parent DNA, now you have two DNA. Then from two, when you repeat this cycle multiple amount of times, from two you will have four. Then from four you will have uh, uh, eight, and so on. So that is how these are the different steps of PCR. Here again, there is one image. As I told you, in the first cycle, these three steps are happening of denaturation, annealing, and extinction. You first put the DNA at high temperature 95 degrees Celsius, it will denaturate. It will become two single strands. At annealing around 65 degrees Celsius, the primers will come unattached. Then in extension, Taq polymerase will extend and make, so from 1 you will get 2, from 2 you'll get 4, from 4 you'll get 8, and so on. It will keep on multiplying in the consequent cycles. Here is a small video explaining
Portion that we want to make
PCR.
copies of. We need some kind of buffer to put it in, and then we need things that would be necessary to make more copies of the DNA. So think about what that might mean from our DNA replication video. We'll need primers. Recall that primers help DNA polymerase, a building enzyme, know where to go to start its building. We need DNA polymerase, the building enzyme. Fun fact, the DNA polymerase used is often a heat-resistant type of DNA polymerase as PCR uses heat. Typically, the polymerase chosen for the job is Taq polymerase, a type of heat-resistant DNA polymerase. Taq polymerase is originally from a type of bacteria that can handle and live in really high temperatures in nature, in hot springs. Pretty cool. We also need DNA nucleotides for the DNA polymerase to build with. So, we can look at the PCR sequence in 3 major steps. We'll illustrate with one double-stranded DNA molecule here, and let's assume this is what we want replicated. Step 1, denaturation. You've heard that word denature before, right? When we were talking about enzymes? One way we had talked about denaturing enzymes was using heat, and heat is what we will use in this step. This step involves the addition of heat needed to separate the 2 strands of the DNA molecule. Step 2, annealing. Okay, so this word means something a little different in biology. Basically, this is when the 2 DNA strands that now have been separated by that heat are going to be cooled and be joined by the primers. The temperature for this step should allow the primers to bind to the specific segment of DNA that you want to amplify, which means make copies of. Step 3, DNA synthesis. Remember, synthesis means to make something. With DNA synthesis, we're going to make more copies of DNA. And to do so, DNA polymerase will begin to work on both of these strands, and it will use the DNA nucleotides as its building material to amplify the DNA. We should note that the temperature at this step may be a little warmer than the previous step. It needs to be a temperature that is ideal for the specific DNA polymerase used. Now, after finishing one cycle of this, you have 2 double-stranded DNA molecules, right? Similar to how it would be in DNA replication within a cell. But you can repeat this now, except this time you now have 2 double-stranded DNA molecules to start with. So you repeat with the denaturation, annealing, and DNA synthesis steps. Now you have 4 double-stranded DNA molecules. You repeat the steps again. Now you have 8. And if the process is automated by a machine, which, why yes, they do have in science catalogs, you can actually do this fairly quickly.
So this is how PCR works. Now, coming back to molecular cloning, after having discussed all of this, I would like to again brief you about what basically is molecular cloning and what are the steps happening. So, this here is your target DNA where the gene of interest will come from. So, basically, it's the cell with the DNA containing gene of interest. From here, you'll isolate DNA. After isolating DNA, you'll get your gene of interest, and gene of, you'll cut it with the same restriction enzyme with which you're cutting the plasmid. As we discussed, plasmid is extracellular DNA. This also you isolate from the bacterial cell and you cut the plasmid with the same enzyme, and then both of them will ligate because they'll have complementary sticky ends. And then now you have recombinant plasmid, and from the recombinant plasmid you'll then transform it into a bacterium, and then these bacteria will be allowed to reproduce and multiply, and then you'll have multiple copies of the recombinant DNA. Isolate gene of interest, insert the gene of interest into suitable plasmid. I introduce the recombinant plasmid into the host organism, select the transformed host cell, and you multiply the introduced gene from the host cell. So, this was all about molecular cloning. The next lecture was about PT cotton. The next few lectures were about PT cotton. And there was a lecture in between in which they actually showed you a hands-on how to perform a PCR and how to run an agarose gel. So you might as well go and have a look at that hands-on training session, because that will be helpful if you have to conduct lab experiments. So talking about BT cotton, so talking about cotton, you might all be aware about what is cotton, because all of us wear this fabric on a daily basis. All of our clothes actually contain some percentage of cotton in them. So cotton basically comes from the genus Gossypium and comes from the family Malvaceae. Other plants belonging to this family Malvaceae include okra, which is bhindi that we eat, or lady finger, known in some basic English. So that's okra. There's hibiscus, that's the flower, which is very famous. And then there's mallow. So these are some other plants that belong to the same family, Malvaceae. And you can see here, most of these plants actually have a very similar flower structure, and they have these lobed leaves. So all of them have these leaves which have these three lobes, or they can have multiple lobes, and um, the amount of cut in the leaves can also be different in different plants, but they all have lobed leaves, and they all have a similar flower structure with this darker and lighter pattern in the flowers. So this is about the phylogeny of cotton. Then moving on to agricultural cotton, from which we actually get the cotton fiber, that is derived from four species, which is Gossypium hirsutum, Gossypium arboreum, Barbadens, and Herbaceum. And this is what the agricultural cycle of cotton looks like. So basically, it spans 5 to 6 months. In the first 2 months or 2.5 months, what is happening is, it from the seedling, it comes to the first flowering point. So from cotton seeds, if you plant them and the plant grows, in the first 2.5 months, it will reach the first cotton bud. In the next 2 months, it will start flowering, and then in the next you will see the cotton boll. So then you will, after 20 more days, you will see the boll ripening. So that is how it's a 5 to 6 month cycle. And as you might already be aware, India is one of the biggest producers of cotton in the world. Cotton agriculture in India, so it requires a period of so for cotton to grow, the plant requires a period of a certain period of frost free growth. So it should not have any frost on itself, the plant, and it needs a lot of sunshine and a dry climate. And it develops, or the plant grows best, when the air temperatures range from 21 degrees Celsius to around 32 degrees Celsius. So between these range, the plant grows best. And there are three main regions in India that grow cotton. The north zone, here you can see Punjab, Haryana, Rajasthan. This is the north zone that grows a lot of cotton. But with the color, you can see they're still less than the central zone, which consists of Gujarat, Maharashtra, and Madhya Pradesh. This is where the maximum amount of cotton grows in India. And then there's the south zone, including Karnataka, Andhra Pradesh, and Telangana. So Odisha also grows some amount of cotton, but these are the three main zones of cotton growth in India. And it grows well in the alluvial soils found in Rajasthan and Punjab, and in the black soils found in Maharashtra. So this is about cotton agriculture and the species as we discussed. So the species, the Gossypium hirsutum, it's actually a cotton tetraploid, as you might be aware of ploidy. So diploid is an organism which has two copies of DNA, and tetraploid, it has four copies of DNA. So the cotton hirsutum is Gossypium hirsutum is actually a tetraploid. Then there is Barbadens, which comes from Egypt. Then there is Arboreum, which is a desi cotton from India, and Herbaceum, which is another desi cotton of India, which is a diploid. These two are tetraploids, Barbadens and hirsutum. So history of cotton in India. Let's talk about how did cotton cultivation start in India? How far does it date back? So they actually, you can even find cotton fibers in the excavations that come from Harappa. So during that civilization, during Indus Valley civilization, also cotton was there. Then, in around 1500 BC and to 1500 AD, India had the monopoly of cotton production industry. So for so many years, India was monopolizing cotton production. Then in 1818, the first cotton mill was established in India. In 1870s, the cotton textile industry expanded in India. Then around the time of uh, just before independence, the Swadeshi movement happened, and it propelled the growth of this industry in India. And then in 2019, now India is still the world's largest cotton producer, accounting for 38% of global cotton. Here you can see a very old hand painted cotton rope which comes from 17th century. So you can see how very delicate and very the amount of handiwork, the amount of effort that goes into making such a outfit is so much. So India used to produce very high quality cotton textiles. Again, there is this early 18th century palampur. It's a wall hanging which is made out of cotton. So this is also, you can see how delicate is the work done in this, how different colored fabrics have been used. Then you can see here, this is what hand ginning looked like. Ginning is actually basically extracting the lint from the cotton seeds. So in India, mostly cotton was extracted using a ginning technique. So you used to use these hand machines. Then what happened is that Indian textiles in that time, they were famed globally, and a huge quantity of textiles were exported. Even all of these travelers which came to India, they used to take huge amounts of Indian textiles and they used to export it. And Indian farmers and weavers, they were very skilled in that time in growing the cotton, in weaving it, in dyeing it, and on the embroidery and the handiwork that we talked about. As you can see from the image, it's very clear. And farmers in those times, they used organic farming techniques, and they tried to reduce vulnerability to pests and improve high yields just by using these organic farmings only and not using pesticides and fertilizers and all of those things. And then that time we had a lot of small scale weavers which used the techniques that were specific to their own location. From where they were, the techniques were also very specific to the location. And these weavers used to clean the cotton, spin the fibers with their own household machines, and they used to make cloths of varying fineness depending on the end where it's going to be used. So if they're making it for high gentry, they were using very uh, high quality fabrics and things like that. So the history of cotton goes forward. Basically, after this, the East India Company came to India, and they used to import large quantities of cotton textiles from India and sell them in Europe in high prices. But what happened was the colonial era. It then coincided with the Industrial Revolution, and in Industrial Revolution, the cotton gin was invented, which used to run on which was not run by hand. It was an automatic machine which used to run on fuel. And it could extract a large quantities of fiber in very less time. So obviously, a machine as compared to a human can do the work better. So it used to extract a large amount of this fiber from the seeds, and this fiber was then made into yarn, and that production was also sped up. And then cotton mills were set up, as you can see. So this is the cotton gin, which used to extract large amounts of fiber. And from here, you can see that cotton mills were set up, which used to convert this yarn into textiles at a very high speed as compared to when it is done by hand. So that's why what happened was the East India Company, now since they had a technique of making textiles very fast as compared to how Indian producers were making it. So they stopped importing textiles. They now started importing raw cotton. So the import of raw cotton, it increased huge amounts compared to the import of the cotton textiles. So what happened was, now the Britishers, now since East India Company, they started importing huge amounts of cotton. One thing that they wanted was that the cotton they import actually should have a longer staple length. Staple length, you can see here. This is the staple length of different cotton species. So you can see herbaceum and barbadans have smaller staple lengths as compared to hirsutum cultivated one. Don't compare it to the wild ones. Wild of all of these have lower what do you call it, have shorter staple length. Staple length is the length of a bundle of fibers. So the wild varieties have smaller staple lengths, but the cultivated varieties have longer staple lengths. And in that also, hirsutum and barbadans have longer staple lengths than herbaceum. As we discussed, herbaceum is diploid, and these both are actually tetraploids. So they have a longer staple length. So what happened was, for these cotton gins to work efficiently, since they were faster, they required a longer staple length. And again, the cotton mills also required a longer staple length for them to make textiles. So what now Britishers were looking for, what East India Company was looking for, is to find colonies in their under them which could produce the longer staple length fabrics. But in Africa, you found barbadans, but hirsutum was found in USA, and from there they used to import large quantities of hirsutum. But in India, we only had herbaceum and the other herbaceum and arboreum. In India, we only had these two desi cotton varieties, herbaceum and arboreum. We didn't have hirsutum. But later, what happened was that USA had a civil war, and then they stopped their export to Britain completely. And now the East India Company was looking for other colonies to grow this hirsutum variety, and they tried to introduce hirsutum into India. And they introduced this into India, and where they were trying to grow it in India, but it was not. All of their efforts were to no avail. These hirsutum varieties are not able to grow in India, of course, because USA, if you look at their temperatures, it's a subtropical country. But India is more or less, it's more warmer. The soil is different, so all of these conditions matter. So they're not able to grow this hirsutum variety in India. But uh, then what happened was, after independence, the government started focusing a lot on cotton agriculture, because India was exporting huge amounts of cotton, and that is where a lot of revenue came from. So the government also started focusing a lot on this cotton agriculture, and they made it a very capital intensive project. So a lot of capital was invested into cotton agriculture, and they started introducing these hybrids. And some of these hybrids, like the Dharwar hybrid of the hirsutum, actually started growing in India. So they introduced these hybrids, and they used a lot of fertilizers and pesticides there. That use also increased. So that is also where a lot of capital went in India. So this was about the history of cotton in India and how these different varieties are now grown in India. Let's talk about the pests for the plant of cotton. So pests, actually, you call an organism a pest only if its actions have become detrimental to human economic concerns. Otherwise, if you have plants in the forest and they are getting impacted by certain insects or certain larvae, you don't call them pests because you don't care about it. You call it pests only if it is affecting the human economic concerns. There are two types of pests in cotton. One of them are sucking pests, which actually suck the sap from the leaves of cotton, uh, decreasing the health of the plant. And the other ones are bollworms, which directly affect the cotton bolls, which we are interested in. They consume the cotton bolls, they eat the cotton bolls. And that's why they're the primary pests, because they are affecting these cotton bolls, which we are primarily interested in. And these pests, there are three types of these bollworms: American bollworm, spotted bollworm, and pink bollworm. But in India, we majorly, our cotton plants were affected by American bollworm, which is Helicoverpa amigera, and this pink bollworm, which is Pectinophora gossipila. So we were concerned with these two bollworms. And the pests, they impact the cotton yields, and the pesticides were then developed to reduce this impact, but the pests were then able to develop resistance against these pesticides. So that is a cycle with pests which always happens if there is this evolutionary race between things. So if you have pests which are impacting your yields, humans then develop pesticides, they spray the plants with pesticides, but the pests will also then mutate, and they will develop resistance to these pesticides, and you need to keep on developing newer and newer formulae for these pesticides. Coming to polyploidy in cotton, so you have this Gossypium, which is the genus, and there are other diploid plants, and there is this plant which is the Raimondi, which has this diploid gene of DD, and then there is Herbaceum, the wild Herbaceum double A, and then they both come together and they form this polyploid. They hybridized and form this polyploid, which is called Eucatens, which is double A double D. And then from here, Herbaceum has also been domesticated, and you can again see that the fiber length is much longer, the staple length is much longer for the domesticated variety. So this is how polyploidy happens. It can happen in the wild itself, but then it gets domesticated by humans because the quality of those are better. That's what happened with wheat also. Wheat is also a polyploid of the wild wheat varieties that are found. Fine. So coming to bt cotton, before we go to BT cotton, we need to be aware about this bacteria which is called Bacillus thuringiensis, which is where the BT comes from in BT cotton. It's a soil-dwelling bacteria, and it has two kinds of states. It has a vegetative state and a spore state. In vegetative state, it grows normally, but in the spore state, it makes the spores when the climatic conditions are not very good. And what we are concerned with is the spore stage. In the spore stage, it has this crystal. These crystals are there present in the bacteria in the Bt spores, which contain this Bt toxin. So these crystals have the cry protein, which is also called the Bt toxin, and it can cause the death of many insect species. And it is used as a pesticide because these insects are actually impacting your plant. So you use this bacteria to your advantage and use it as a pesticide to kill the bacteria or kill the insects pests. So this is because Bt has spores, and these spores contain these crystal proteins, which are called cry proteins for short. And these, when they are consumed by the insect larvae, if the insect larvae is consuming a plant which has been sprayed by, say, these Bt spores, if that consumes it, it is lethal because these crystal proteins, they get lodged into the alimentary canal of the insect, and then they start multiplying, which leads to the eventual death of the insect. So what was done initially was these sprays were made, pesticide sprays were made, which contained the Bt spore. And this was the plants were entirely sprayed with these Bt pesticide, and that was enough to actually kill the insects. But then this could also, and it is helpful because these crystal spores are very very specific to their target. So if they are impacting certain insect species, they'll impact those only. They'll not impact the good insect species which are actually helpful in pollinating the plants, and they'll not impact humans as well. So that is why um, it was a good pesticide. You could spray it over the plants. Then researchers moved forward and they said, okay, we can actually just introduce the gene for this toxicity, for the bt toxin. We can actually just introduce this gene into the plant, and then we don't need to spray the plant with the pesticide because the plant itself will have the toxin. So they then what happened is, then they carried out this recombinant dna technology, which can be done in plants. So for this, what you need to be aware of is this bacteria called Agrobacterium tumefaciens. Agrobacterium tumefaciens is actually a bacteria which causes galls, which causes this crown gall formation in plants. So it negatively impacts plants actually, and it contains this plasmid with tDNA, and this is like a transfer dna, and it can actually it leads to multiplication of the plasmid and death of the plant cells. So but we use this agrobacterium tumefaciens to our benefit. What we are doing is, we actually isolate this plasmid from the agrobacterium tumefaciens, and to this plasmid we cut it with the restriction enzyme, and we add our gene of interest, and we take down the toxicity genes which can actually harmfully impact the plant. So number one, you treat foreign DNA and plasmid with restriction enzyme. So you treat them with the same restriction enzymes, and then you treat them with DNA ligase, which will lead the gene of interest to get ligated into the plasmid. So now you have a recombinant plasmid. Then you introduce the recombinant plasmid into the cultured plant cells. So you have a cell culture of the plant cells, in which you will now introduce this recombinant plasmid. So when you have this recombinant plasmid, which will then insert it into the plant chromosome. So here you can't have extra chromosomal plasmid because in eukaryotes it does not function that way. So you need to introduce it into the chromosome of the plant. Then you regenerate the new plant which has with these cultured cells. So this plant will now carry the trait which of the gene of interest. So it will now be expressing the gene of interest. It will now carry the trait. So in this case of bt toxin, we wanted it to carry the trait of this toxicity. So that is how cotton was actually made into recombinant bt toxin cotton. Then comes the discussion of whether bt cotton was good or bad for India. So there are two views about this. One view, which comes from environmental activists and from NGOs, it exists, which says that it was a failure. It was an abject failure. It led to cases of crop failure. There is a link between BT seeds and farmer suicide because BT seeds are so expensive that farmers cannot afford it, which led to a lot of farmer suicide. And the timing and sustainability of BT cottons positive impacts actually overlaps with many other things, and that's why it's a bad. But if you look at how comment the economists and how the industry supporters comment, they actually think it has been a technological triumph, and BT cotton has been very nice. And there has been the adopters that have taken up BT cotton in their fields have benefited much more from it than people who have not taken up BT cotton in their field. So you can actually go on the I Think Biology textbook and study more about BT cotton. What was actually found in a study later on was that. The success of cotton agriculture cannot all be attributed to BT cotton because even though BT cotton was introduced, you can see that cotton yields have actually increased with, and they follow a pattern of increase with the increased amount of use of fertilizers and irrigation. So since more fertilizers were added to the fields, more and more irrigation happened, and even in Gujarat, because of the Sardar Sarovar Dam, the irrigation increased, and that is when cotton yields also hiked. So cotton yields also increased when irrigation and when fertilization increased. So you cannot give all of the credit to BT cotton. Another thing was that even though a lot, like 90% of land in Punjab, Gujarat, Rajasthan was under BT cotton, the cotton yields were not increasing. They increased only after fertilization and irrigation were added to the mix. So that is why you cannot actually give all of the credit to BT cotton when it comes to improved cotton agriculture in India, because there are other factors at play which you cannot oversee. So this was all about BT cotton, and this was all that was discussed during this week. And now we have a small Q&A session, uh, which will have 15 MCQ questions, which will test your understanding of what happened during the week, and which will help you during your exams. So the first question is, so let's read the following statements and select which of the option is correct. First statement says, endonucleases are used to cut DNA at desired sites. Okay. And the second statement says ligases are used to join separate DNA fragments together. So we have four options here. Either statement one is correct, but statement two is incorrect, or statement two is incorrect and statement two is correct and one is incorrect, or both the statements are correct, or both the statements are incorrect. So let's think about it for some time and then see what will be the answer to this question. So the correct answer to this question is that both the statements are correct. As we've already discussed, endonucleases do actually cut the DNA at desired sites when they recognize palindromic sequences, and ligases are actually used to join the DNA fragments. So that is what we use to ligate the recombinant to make the recombinant plasmid by ligating the host plasmid and the target gene of interest. Okay, moving on to the next question. So the next question says that new technologies have to be evaluated carefully. So which of the following is true? Number one, cotton production in India increased overall around the same time that BT cotton was introduced. Okay. Second statement, while pest resistance against lepidopterans was seen after BT cotton was introduced, farmers had to spend more money on pesticides against other pests. We witnessed, third statement, we witnessed a decrease in fertilizer use, pesticide use, and overall cotton plantation acreage since BT cotton was introduced. D is all of the following, all of the above are true. And option E says A and B, these two statements are true. So, which of the following is correct? Let's think about it sometime and then I will let you know the answer to this question. So, the correct answer to this question is that both A and B, the two statements are correct because cotton production in India did increase overall at the same time that BT cotton was introduced. And that is why I was saying you should not give all the credit to BT cotton because there were other factors at play as well, which were not considered properly. And while pest resistance against lepidopterans, which were these gallworms, these bollworms, it did increase because of BT cotton, because of the toxin being inside the cotton. But yes, since there is a niche which was, so you know about biology, if there's a niche which has been emptied, so since there are no longer lepidopterans there to affect the cotton bolls, the other pests, which were the sap-sucking pests, they increased tremendously. And that is why the farmers had to spend more money on pesticides against other pests, even though the lepidopterans had decreased. Next question. Which of the following is not true about cry or crystal genes that produce crystal proteins as pesticides? So, which of the following is not true? Number one, there is a high level of specificity in terms of the pests that the toxins affect, minimizing off target effects on other beneficial insects. Insects stop feeding within a few hours of ingesting the cry protein, saving the plants from damage. The toxins have known effects on human health, or none of the above. So, which of the following is not a true statement? Think about it for some time and then I will let you know the answer. Yeah. So the correct answer to this question is that the toxins have known effects on human health. This is the incorrect statement. As discussed, since only because these toxins were known or were found to have no effects on human health were they used as a valid pesticide. So they have a high level of specificity, as I already mentioned, and therefore they only affect the terms, the pests that we want them to affect, and they do not affect other beneficial insects. Another thing is when an insect feeds on a plant that has been sprayed by BT cotton or that actually itself has carries the BT toxin, it stops feeding within a few hours, and then it this saves the plant from any damage. Okay. What are some features of a hybrid cotton strain one might select for commercial purposes? So, when you're looking for selecting a cotton strain for commercial purposes, what are some features you would look at in that hybrid cotton strain? Number one, improved bearing capacity, high productivity, continuous flowering, and high yields. Number two, pest vulnerability, less frequent flowering, and stable yields. Number three, greater flowering height, increased water requirement, and pest resistance. And number four is none of the above. So, what do you think will you look for when you're looking at a cotton hybrid strain? And what of the features will you will you select while choosing a strain? So the correct answer, as you might have already guessed, is improved bearing capacity, high productivity, continuous flowering, and higher yields. So you would want plants that have high bearing capacity. They bear a lot of cotton bolls. They should have a high productivity. The cotton bolls should have high amount of cotton in them. They should be flowering continuously so that you can get more and more cotton bolls, and there should be higher yields. Okay. What are the features of the domesticated species of cotton that we harvest today? So, the cotton that we are harvesting today, the domesticated species, what are its features? Does it have improved coloring? Does it have a longer fiber length? Does it have double duplicated genome leading to polyploidy? A and B, or B and C only? So, what are the features of domesticated cotton species that we are using today? So, the correct answer to this question is B and C, as you might have already guessed, because we looked at longer fiber length, and so it has a longer fiber length as we discussed, and it has they are tetraploids, so they have double duplicated genomes. Okay. Number six, restriction enzymes recognize and cut DNA where? At random sites, at palindromic sequences, at AT rich region, or at start codons. Where do restriction enzymes function? Where do they recognize DNA and where do they create a cut? So the correct answer to this question is at palindromic sites. As we already discussed, when the sequence reads the same from 5' to 3' on both the DNA strands, that is where the restriction enzymes recognize and cut the DNA. What type of ends do restriction enzymes produce? Do they produce sticky ends, blunt ends, both A and B, or only blunt ends? So when you cut a DNA molecule with a restriction enzyme, what kinds of ends can it produce? So the correct answer to this question is both A and B. It can produce either sticky ends or blunt ends. So what are plasmids? Are they small circular DNA molecules in bacteria? Are they proteins involved in recombination? Are they RNA molecules used in translation, or are they lipid molecules found in the cell membrane? So what really are plasmids? So the correct answer to this question is that plasmids are small circular DNA molecules in bacteria. We have discussed this at length now. What is recombination? Is it a process of breaking and joining DNA strands? It is synthesis of proteins from RNA. Is it production of our restriction enzymes? Or is it separation of DNA fragments? So, what is recombination? Breaking and joining DNA, synthesis of proteins from RNA, production of restriction enzymes, or separation of DNA fragments. The correct answer to this is that recombination is actually the process of breaking and joining DNA strands. So, as we know, we use restriction enzymes to cut up the DNA strands, and then we use ligase to join the DNA strands. What is the role of Taq polymerase in PCR? So, what does Taq polymerase do in PCR? Does it cut DNA into fragments? It binds to the promoter region. It synthesizes new DNA strands, or it acts as a restriction enzyme. So, what is the role of Taq polymerase? Yeah, so the correct answer is that it synthesizes new strands of DNA. So it acts as a it is a DNA polymerase and synthesizes new strands of DNA. What is the purpose of gel electrophoresis? To amplify DNA, to cut DNA at specific sites, separate DNA fragments based on size, or to transcribe RNA. So what does this lab technique, gel electrophoresis, do? Yeah, so the answer to this question is that gel electrophoresis is actually used to separate out different DNA molecules based on their size. 12th question, BT cotton is genetically modified for what? Does it resist drought conditions? Does it produce its own insecticide? Does it have a longer fiber length, or does it grow faster? So, what is the correct answer to? So, what is the correct answer to uh, BT cotton? So BT cotton actually, as you might be aware by now, it produces its own toxin. So this is this BT toxin which we get from bacteria thuringiensis, bacterium, Bacillus thuringiensis. From that, we actually want the BT toxin to be there in the plant itself. So the plant itself can produce its own uh, insecticide. So the Bt gene, oh, sorry, I gave away the answer in the last question. The Bt gene in Bt toxin is derived from what? Is it derived from a plant virus? Is it derived from Bacillus thuringiensis, the bacteria? Is it a cotton plant mutation or is it a fungal pathogen? So, where does the Bt gene come from? So, I've already given away the answer to this question, and the correct answer, as you might have already thought of it, is the Bt comes from Bacillus thuringiensis, the bacteria. Question number 14. Which of the following is not required for PCR? So, which of these is actually not a requirement for PCR? Is it the DNA template? Is it the RNA polymerase, primers, or nucleotides? Which of these is not required for PCR? So, yeah, the correct answer to this question is RNA polymerase. Why would we need an RNA polymerase? We would actually require a DNA polymerase for duplicating the DNA templates. Why are plasmids useful in genetic engineering? Why do we use plasmids in genetic engineering? Can they replicate independently? Do they carry antibiotic resistance genes? Can they be modified to carry foreign DNA? Or is it all of the above? All of these statements, are they all correct? So, what makes plasmids useful in genetic engineering? Yeah. So the correct answer to this question is all of the above. Plasmids can replicate independently of the host chromosome. They do carry antibiotic resistance genes and they can be modified to carry foreign DNA. So that was all for today's class. Thank you for listening. If you have any doubts, you can type them out in the chat box and I will take a look at them and try to answer you. Do take a look at the lectures of this week present on the NPTEL course page. And yeah, see you next Saturday. Thank you.